Hello, this is our LB-145 final talk, and our group name is the Mutation Busters.
Okay, our introduction, our 2869 INSG mutation is our mutation. And basically, what our purpose is to design a PCR test or an assay to detect the presence of the CFTR mutation of our 2869 INSG. And what our hypothesis is by creating a diagnostic assay for patients with the 2869 INSG mutation and cystic fibrosis using optimal temperatures and times of PCR through accurate measurements of concentrations that a single base pair insertion will detect the 2869 INSG based on prior diagnostic testing, Gasparini 1992. And what our experimental design is that we created design mutant primers, and we'll use a methodology of polymerase chain reaction and gel electrophoresis. And what PCR is that it amplifies the DNA by combining our DNA, our templates, our Taq polymerase, and our primers we decide to use. And gel electrophoresis is a process where we create a gel using buffer and agarose. And what this gel does is as it separates the DNA bands, and it locates the placement of the base pairs, and we can see where they are um, using UV light and measuring the bands and confirming where the spots are. And this methodology does work because of our process to confirm where the lambda was and as a positive control. And in the future, our published primers, which will also serve as another positive control. And this is significant in science because these tests will help doctors diagnose cystic fibrosis patients with a specific mutations like 2869 INSG, as it is an uncommon mutation in cystic fibrosis. There's not many papers already or many tests done to confirm the presence. So our processing of creating our own tests will allow doctors to diagnose this mutation in more of an efficient manner.
This figure shows the stepwise process of DNA extraction from human spit, which shows the progression from buccal cell collection to purified DNA using Killick resin. So there's a long procedure that involves many steps in this. So the purpose of this procedure is to isolate and purify DNA for applications such as PCR. The process basically begins with the collection of human spit, well, around 1.5 milliliters, which contains buccal cells. The sample is then centrifuged to separate the supernatant from the buccal cell pellet. Helix resin is then added to that pellet, and the mixture is heated to lyse the cells, which releases the DNA. Following that, another round of centrifuge happens where the helix resin and cellular debris are separated, which leaves the purified DNA. The final result is a clean DNA sample, which is suitable for molecular analysis, and that's done using nanodrop technology. So there's two figures here. Figure two is the lambda DNA gel electrophoresis results, which is the left, and a corresponding standard curve on the right shows a lambda virus band migrating 2.929 centimeters from this well, which corresponds to approximately 346 base pairs as determined by the intercept point on the standard curve. This figure basically demonstrates the process of determining the size of a lambda virus DNA using gel electrophoresis and a standard DNA ladder. The purpose of the analysis was to estimate the number of base pairs in the lambda virus fragment by comparing its migration distance of that of known DNA standards. This method involved loading the lambda virus DNA and a SID Labs 100 base pair DNA ladder into a gel running electrophoresis to separate the fragments by size, and measuring the migration distance from the wall to the band of interest. The lambda virus band migrated 2.929 centimeters, as you can see here and here. And this value was plotted on the standard curve generated from the ladder distances and their corresponding base pair sizes. Using the best fit logarithmic equation from the curve, The DNA fragment was estimated to be approximately 346 base pairs in length. Figure 3 shows a diagram of a primer binding sites on the lambda DNA sequence, illustrating a 395 base pair region amplified by the forward primer and the reverse primer. The purpose of this design is to target a specific DNA segment for amplification using polymerase chain reaction, also known as PCR. The forward primer binds to the bottom strand of the 5 to 3 direction, while the reverse primer binds to the top strand in the 3 to 5 direction. These primers attach to the target sequence, enabling selective amplification of the 395 base pair region of PCR. Successful binding of both primers ensure the specific efficient replication of the desired DNA fragment, which can be analyzed in gel electrophoresis.
So here's our figure 4. It's a gel electrophoresis analysis of our published wild-type CFTR gene, which was amplified via PCR from the human genome. So we have the 100 base pair plus DNA ladder, and all the base pairs are labeled according to the bands that showed up. And then the 8 different wells, we used our published primers that had different annealing temperatures, which are also labeled accordingly. And we were expecting at least one of the primers to show up on the gel at 468 base pairs. But as you can see, the furthest one only went to about 200. So this is an example of a failed gel. To the right is a graph showing the base pairs and their corresponding migration distance. So say the gel was actually a success, the band would have showed up at about 468 base pairs with a total migration distance of about 2.75 centimeters. And this is figure 5. And here is our depicted mechanism of published primer binding sites. To amplify a specific piece of DNA using PCR, we use 2 primers, a forward primer and a reverse primer. The forward primer binds to the start of the region we want to copy. It attaches to the bottom strand, but it matches the top strand because primers are always written 5' to 3'. And the reverse primer binds to the end of the region we want to copy. It attaches to the top strand, but is the reverse complement of that part of the strand. So in this case, the DNA is 486 nucleotides long, and the primers define the start and end points for copying. The arrows in the figure show that both primers face inward, which is essential for making the full copy.
Okay, moving on to figure 6. What this figure is a mechanism of just any frame shift mutation in the gene sequence, which in the future, the 2869 INSG mutation is a frame shift mutation. So this is a mechanism representing how it generally works. So that was the purpose. And the way the mutation works is that on our top 2 primers, this is like your wild type forward DNA sequence. You have a primers running 5' to 3', and then you have your reverse or your complementary 3' to 5'. And as you can see here with the green nucleotides, they all bind to each other correctly. But when we look to the right, the insertion of any nucleotide causes a shift in the primers in the gene sequence. So as you can see depicted by the G in the red arrow, if you insert a nucleotide, everything shifts. So now when you go down to the now mutant type 4 DNA sequence, as you insert that nucleotide, your forward primer will all the nucleotides will shift over one, causing your complementary not able to bind to their corresponding nucleotides. As you know, A binds to T and C binds to G. But now adding that G highlighted in yellow, now you have nucleotides that won't anneal correctly. So now since you added it in, everything shifts over right to the one. So everything highlighted in red from the right, depicted by the blue arrow in the frame shift mutation, your primers now won't anneal, meaning that the base pairs won't appear. So moving from the G right, you have A to C, G to A, and T to G. And what the attack does is it's not going to build its nucleotides onto the primer since they don't anneal correctly. So when a frameshift mutation occurs, those nucleotides won't be present, causing this frameshift mutation to not work, and end up finding that you have the mutation and your proteins won't fold correctly. Now in figure 7, this is a mechanism of our design primers via a frameshift mutation in the 2869 INSG. So the figure prior was just an example. I was representing a mechanism using random nucleotides. Specifically in this one, this is in the CFTR gene sequence. This is where, like, since our mutation is an insertion of the gene nucleotide at the 2869 spot, this gene sequence is around like 2800. So as you can see here, we designed our primers based on where it was located so we can detect the gene. So in our 2869 INSG forward wild type DNA sequence, verbatim from the CFTR gene sequence, we have 20 nucleotides that are supposed to anneal correctly. And then what happens at the 2869 position is that G, like our mutation says, insertion of G right after the 2869 position. So right in between 2869 and 2870, there's an insertion of the G nucleotide causing this frame shift. And the way we create our primers is that we made it so the ends where they anneal they can't keep annealing. So like when a frame shift occurs, most of them won't anneal correctly, but if you let the gene sequence go on, some of them will pair up, as you see here at the end of our 2869 INSG forward mutant type DNA sequence. At the end of our complementary, the G and the C, they do bind together. So we carefully, specifically picked out primers where most of them won't anneal correctly. So, and we have a universal reverse 2869 INSG primer that where it's from the wild type DNA sequence. This primer is supposed to be the band's supposed to be placed at 200 base pairs, while as we on our designed mutant primers, since everything highlighted in red, again, like I said before, they don't bind correctly. Those nucleotides will be left out of the your base pairs. So we predict that after an insertion of G, 11 of these nucleotides won't be present. And so that means that when we run a gel, that it should appear at 189 base pairs because of the shift of the G placed at the right after the 2869 position of the primer that runs 5' to 3'. The complementary 3' to 5' won't be able to bind correctly. And we'll end up finding that your wild type primer DNA base pair band will appear at 200, and your mutant type using mutant DNA will appear at 189 base pairs using the universal reverse. And that's what happens when this is just a mechanism showing all of that and the insertion of the G and what will happen to the sequence afterwards. Every nucleotide will shift, shown by the arrow right under the forward mutant type DNA sequence. And so those won't anneal correctly. Now, moving on to figure A, this is a predicted gel outcome for our design primers. So in well 1, we have our 100 base pairs gold bio MW ladder, and depicted by the bands, we have all these bands labeled by the letter. And well 2, we have our wild type cystic fibrosis DNA, paired it up with our buccal cells. And this, we predicted in our design primers, it will appear at 200 base pairs. Well 3, we have our mutant type cystic fibrosis DNA, and that will show up at 189 base pairs. And the homozygous cystic fibrosis DNA in well 4 is depicted where if you are a carrier with both alleles, you will have both bands show up at 289. So the purpose is to predict our gel outcome, and this is what we think it will look like. And the methods behind all of this is that we'll run two different types of PCR. We will run PCR with equal amounts of water, DNTPs, Taq polymerase, and then for our wild type PCR cocktail, we'll have 1 microliter of our forward and reverse of our wild type and 1 microliter of buccal cells. Compared to our third well of mutant type, we will have 1 microliter of our forward and reverse primer of our mutant primers we designed, depicted by the figure prior, with mutant DNA rather than buccal cells. Because if we did with buccal cells, then nothing would show up because they don't have the mutation. And doing so, we will run another PCR. Both the mutant type and the wild type have the universal reverse, but we ran it with in our fourth well with a homozygous carrier. They will have both bands show up, and that's for PCR and gel electrophoresis. What we'll do is we'll make a gel using 0.5 grams of agarose powder, water, and buffer to make a gel, and then we will run it using gel electrophoresis, and then we will see the results looking in the UV light. And based on the UV light, this is what the gel should look like. So the green bands are the wild type, the red bands are the mutant type, and that we predict all these results will show up. And yeah.
Thank you so much for listening to our presentation.